Hello everyone, welcome to Brain Blitz Audios. In today's episode of NEET Preparation, we'll be dealing with previous year questions of biology in medical entrance examinations in India. The chapter that we'll be dealing with is the 10th chapter in the grade 11 syllabus for CBSE. The name of the chapter is Cell Cycle and Cell Division. Now this is a really short chapter and there aren't many questions asked, but they are very important. So please make sure that you watch the video completely in order to understand the most important questions occurring at the end. So let's begin, shall we? The correct sequence in cell cycle is S, G1, G2M, SM, G1, G2, G1S, G2M, M1, G1, G2, S. So which of these is the right option? Let's look at a sequence of the cell cycle. So there are 4 stages. In the first stage, the centromere is formed and the cell develops. And then the DNA replicates. And after that, the accessory proteins form for cell division and the cell develops further. And in the end, we have the actual cell division. So that is the sequence of the cell cycle. So centromere is duplicated first, then DNA replicates. After that, we have protein synthesis, as well as the further development of the cell. And finally, we have the actual cell division. So cell division is mitotic or meiotic, and it's represented by M. And the centromere formation is part of the interphase. It's the first part of the interphase known as G1. Proteins are formed in G2, and DNA replicates in the S phase. So if you look at our sequences in option A, S comes first. So that means it says that the DNA replicates first, but in reality, the centromere replicates first. So option A is incorrect. In option B, it is the same scenario because S comes first. So they're saying that DNA replicates first, which is incorrect. In option D, M comes first. So that means mitosis happens first before the preparation for mitosis, which is absolutely wrong. So option D is incorrect. The right answer is option C, G1 to S to G2 to M. So that follows the correct sequence. Centromere duplicates first, then DNA replicates, then proteins are formed, and further development of the cell occurs. And finally, we have the actual cell division. Option C is the right option. Next question. Colchicine prevents the mitosis of cells at which of the following stage? Whether it's A, Anaphase, B, Metaphase, C, Prophase, or D, Interphase. Let's look at what is colchicine. So colchicine is an alkaloid, and it is found in Colchicum autumnale, which is a plant. And this colchicine is referred to as a mitotic poison. It prevents mitosis because of the fact that it arrests spindle formation. So when spindle formation is arrested, it means that the chromosomes cannot be differentiated into the newer cells. And this process of spindle formation happens in the late prophase. So keeping these in mind, let's look at the following options. Option A, anaphase. In anaphase, the actual separation of the chromosomes happen. So by this time, the spindle has already formed. So spindles are present. So option A is incorrect. Option B, metaphase. In this phase, the chromosomes are aligned at the equator using the spindles. So therefore, option B is also incorrect. Option C, prophase. Since spindle formation happens in the late prophase and colchicine prevents spindle formation, option C is the right option. If we choose option D, then it is not present in mitosis. In interphase, no mitosis occurs, but the preparation occurs. So option D is incorrect. The right answer is option C, prophase, because it is in prophase that spindle formation occurs due to microtubules, and colchicine prevents spindle formation, which leads to prevention of mitosis. Next question. Spindle fibers of mitotic cells are made up of tubulin, actin, myosin, collagen. Let's look at each of our options. Collagen is a protein found in connective tissue, such as tendons and ligaments. So option D is incorrect. Options B and C, actin and myosin. Both of these are muscle fibers. So therefore, options B and C are incorrect. Therefore, the right answer is option A, tubulin. Spindle fibers are made up of microtubules, and these microtubules are made up of tubulin protein. So therefore, option A is the right option. Next question. When synapsis is complete all along the chromosome, the cell is said to have entered a stage called as zygote, pachytene, diploide, or diakinesis. So the end of synapsis stabilizes chromosomes, and it also leads to the formation of bivalents or tetrads. So that means two chromosomes with a centromere are connected together. So they become sister chromosomes. So that is what synapsis is all about. Let's look at in which state synapsis is complete, whether it's zygote, pachytene, diploide, or diakinesis. Diakinesis is the final stage of prophase 1 in meiosis. So over here, what happens is that the nuclear membrane breaks, and the formation of chromosomes happens way before the nuclear membrane breaks. So option D is incorrect. Option C, diploide. In diploide, complete crossing over of DNA is occurring, and it also results in the formation of chiasmata. And in diploide, the bivalents separate. So anyways, this stage is past the stage for synapsis. So option C is incorrect. What about option B, pachytene? In pachytene, the main process is genetic exchange between non-sister chromatids. And in order for this to occur, synapsis has to be complete. So option B is incorrect. The right answer is option A, zygote. Zygote is when synapsis becomes complete and the formation of bivalents and tetrads occur, which stabilizes chromosomes. So therefore, option A, zygote, is the right stage where the cell has completed synapsis all over the chromosomes. Next question. Which one of the following precedes reformation of the nuclear envelope during M phase of the cell cycle? decondensation from chromosomes and reassembly of the nuclear lamina, transcription from chromosomes and reassembly of the nuclear lamina, formation of the contractile ring and formation of the phragmoplast, formation of the contractile ring, and transcription from chromosomes. So in these options, one of them is the right option. Let's find out which. Options C and D have the phrase formation of the contractile ring. This contractile ring is used in cleavage, which is a part of cytokinesis, and that happens after the M phase of the cell cycle. So option D is incorrect, which also means that option C also becomes incorrect. What about option B? Transcription from chromosomes and reassembly of the nuclear lamina. Reassembly of the nuclear lamina takes place before the reformation of the nuclear envelope, but transcription from chromosomes, this occurs in the interphase after M phase. So therefore option B is also incorrect. The right answer is option A, decondensation from chromosomes and reassembly of the nuclear lamina. So after anaphase in telophase, chromosomes start to decondense and the nuclear lamina starts to reassemble. So after these phenomena, the reformation of the nuclear envelope takes place in both cells. So option A, decondensation from chromosomes and reassembly of the nuclear lamina is the right option. Next question. During which stages or prophase 1 substages of meiosis do you expect to find the bivalence and DNA replication respectively? So we need to find out which of these forms bivalence and which of these forms DNA replication. Bivalence, in a previous question, we found out that bivalence occur in zygote, and DNA replication occurs in the S phase. This was a part of our first question. So using these two, we can safely conclude that option D is the right option. In options A, B, and C, the first option is packeting. So it says that bivalents are found in packeting. But in packeting, what you find is the genetic exchange of genetic material. And DNA replication is only found in S phase, but in options A and B, you find that it's written interphase. So options A, B, and C are therefore incorrect. Next question. Many cells function properly and divide mitotically even though they do not have A, a plasma membrane, B, cytoskeleton, C, mitochondria, or D, plasmids. So we need to find out which of these is not present in all cells. Option A, plasma membrane, is the wrong option because it is found in all cells which are living. Option B, cytoskeleton, is also found in all cells, so it's incorrect. Option C, mitochondria, is found in one way or another in eukaryotic plants, eukaryotic cells. And it has another counterpart in prokaryotes, so the presence of an energy currency of the cell is pretty important. So option C is incorrect. The right answer is option D, plastids. These are found only in plants. They're not found in animals, But still, animals are able to undergo mitosis, even though they do not have plastids. So option D is the right option. Next question. The stage of meiosis where centromere separates is A, metaphase 1, B, metaphase 2, C, anaphase 1, and D, anaphase 2. In both metaphase 1 and metaphase 2, the chromosomes are aligned in the equator of the cell. So therefore, options A and B are incorrect. The stage of meiosis where centromere separates can either be anaphase 1 or anaphase 2 in meiosis. Let's check which of these is correct. So for that, let's draw the diagram for anaphase 1. So this is a pretty crude diagram. So we have the asters here, and in anaphase 1, the chromosomes are arranged like this. So this is at the start of anaphase. So the spindles are attached to the centromeres, and you notice that this group of chromosomes just parts from this group of chromosomes. However, there is no breaking of the centromere in between them. So option C is incorrect. The right answer is option D, anaphase 2, which has this sort of diagram. Let's draw it. So this is how, in a crude representation, we can find out that anaphase 2 is the phase where the centromeres separate. So one set of chromosomes have gone to one cell, and now you're trying to separate them. So the centromeres are separated in this case, while in anaphase 1, it is not separated. So option D, anaphase 2, is the stage of meiosis where the centromere of chromosomes separate. Next question. During meiosis 1, the chromosomes start pairing at A, leptotene, B, zygotene, C, pachytene, D, diplotene. Pairing of chromosomes occur in bivalents or tetrads. So when we look at our options, in option A, leptotene, it contains the condensation of chromatin material into chromosomes. So option A is incorrect. Option D, diplotene, is where the formation of chiasmata take place and disappearance of bivalents. Option D is incorrect. Option C, pachytene, is where genetic exchange takes place. So by the time a genetic exchange starts to happen, the chromosomes are already paired so that the genetic material between non-sister chromatids takes place. So option C is incorrect. The right answer is option B, zygotene, which contains the stage where bivalents or tetrads are formed. So option B, zygotene, is the right answer. Next question. How many mitotic divisions are needed for a single cell to make 128 cells? So over here, this is a formula-based question. So in the chapter Cell Cycle and Cell Division, you learned that the number of mitotic divisions is given by the formula 2 raised to n. So now we can write 2 raised to n is equal to 128. So 128 on prime factorization. By 2, you get 64. By 2, you get 32. By 2, you get 16. By 2, you get 8. By 2, you get 4. By 2, you get 2. By 2, you get 1. So in the end, we have 1. which cannot be prime factorized further. Over here, you find that there is 1, 2, 3, 4, 5, 6, 7. So 128 is 2 times 2 times 2 times 2 times 2 times 2 times 2, which is basically 2 raised to 7. So 2 raised to n is equal to 2 raised to 7. So therefore, the exponent has to be equal. So n is equal to 7, where n is the number of divisions that take place. So mitotic divisions needed for a single cell to make 128 cells is option A, 7. So you need 7 mitotic divisions in order to make 128 cells. And this is our last option, last question. Match the description given in column 1 with the correct stage of prophase 1 given in column 2 and choose the correct option. So column 1, chromosomes are moved to spindle equator, centromere splits and chromatids move apart, pairing between homologous chromosomes takes place, and crossing between homologous chromosomes. Column 2 contains pachytene, zygote, anaphase, and metaphase. And over here, we have 4 options. Now over here, we just need, if you're smart enough, we just need to find out one of the matchups in order to prove that everything is correct. So let's look at statement A in column 1. The chromosomes are moved to the spindle equator. So when you put that in a diagram in your head, it looks something like this. So the spindles are attached, so they are at the center. And which diagram is this? It is the diagram for metaphase. So therefore, we can write that column 1's statement A from column 1 matches with option 4 from column 2. So therefore, our option must contain A matched with 4. And which of the options contain that? Option C alone. So option C is the right answer. Let's confirm with the other options. It says B3. So centromere splits and chromatids move apart. So, centromere splits like this, and then the chromatids start moving in the other direction. And that is found in anaphase. So B3 is correct. Next, it says C2. So pairing between homologous chromosomes takes place. Pairing happens in zygote, that is 2, so C2 is correct. And finally, crossing between homologous chromosomes, that happens in pachytene because of the bivalent formation, so D1 is also correct. So therefore, just by finding out the matchup between A and 4, we were able to arrive at our complete answer. So these are just some of the shortcuts that people use in order to gain time in medical entrance examinations. So that ends this episode of NEET preparation. So we've done a lot of videos on NEET preparation because we feel that it's better if we do a lot of videos and give detailed explanations about some questions. So if you want to watch those, then please click the links in the description down below that will guide you to the separate episodes and the playlist as a whole. So we also have other useful and interesting content such as mind maps, CBSE board exams, memory hacks, stories, poems, etc. And so all of this happen at Brain Blitz Audios. And so if you want to keep track, please subscribe to our channel and click that notifications button in order to receive updates of our latest content. So until the next episode, take care, stay safe, bye-bye for now.